we will do a small test elucidating the composition of blood.
Composition of blood? How can I show that?
Collect a few ml of blood in a test tube and add a few drops of, that is 1% sodium oxalate or sodium citrate solution to the blood. These salts prevent clotting of blood. Close the test tube with cotton plug. And keep this test tube for about an hour without shaking. A clear, pale straw-colored liquid separates out like this. This liquid is called plasma and remaining mass is called blood cells. Now again collect a few ml of blood in a test tube and do not add sodium citrate or sodium oxalate. And then repeat the procedure as we did before. Do you see the blood clotting in the test tube?
Yes, and also a liquid is separating out.
Good. This liquid is called serum.
You said that the liquid is plasma. Is plasma otherwise called as serum?
No, friend. Both are different. The liquid separating when sodium oxalate added to blood is plasma, and the liquid separating without adding sodium oxalate is serum. The serum does not contain some proteins which are present in plasma. Serum is collected after the clotting of the blood, and plasma is collected without clotting of the blood.
Great, Bob. And I hope I will win a prize for my experiment.